Okay, so factors affecting enzyme activity is going to be the topic of our next lecture. Kind of the bottom line is that evolution has made it so that each enzyme has evolved to work perfectly in its cellular environment that it's found in. Okay, so any changes to that environment, if we took an enzyme out of, for example, a heart cell and put it into a liver cell or something like that, it might not work as effectively. Okay, they're very sensitive to things like temperature, pH, and then the other factor we're going to talk about is the concentration of an enzyme and a substrate. Okay, so first of all, your body goes to great lengths to maintain a specific temperature. And we talked last in chapter 19 about just generally about the effects that temperature has on polypeptide chains. They have a tendency to denature those chains at high temperature. And at low temperature, it's also not really great for the enzymes. They may not denature, but they don't function correctly. Your body is again goes to great lengths to maintain a temperature, what we would call the physiological temperature of 37 degrees Celsius. And so the enzymes in your body are tuned to work to be maximally effective at that temperature. Anything above that point, they'll start to denature, the enzymes just break up. But even below that point, the enzymes do not work as effectively. Okay, so if we just took a simple measure of reaction rate or how quickly how quickly substrates are being converted to products, you would see that and this trend goes for just about every enzyme in your body, it would be maximally effective at that 37 degrees Celsius. Anything higher and the reaction rate will tend to dip off until it goes to zero because the enzyme's completely denatured. And as well as if you go lower than that, you will see a decrease in the reaction rate. There are other organisms that have been adapted to live at extremely high temperatures. So there are certain bacteria that are found in, what are those called? Like Yellowstone, like the geysers and stuff like that, where it's, really, really hot thermal baths, temperatures ranging from 50 degrees Celsius to 120 degrees Celsius. Well, those organisms have evolved to live in those environments. So the optimal temperature for those organisms would be whatever that temperature was, whatever the bath that it evolved in is. So if it's 50 degrees, its optimal temperature is now 50 degrees. Likewise, there are other organisms that have adapted to live at really cold temperatures. And so they would all have maximal or optimal temperatures that at a lower temperature. But for us, for humans, and actually for just about most life on Earth, that optimal, what we would call the physiological temperature, is 37 degrees Celsius. pH. pH is something that's also enzymes are very sensitive to, right? We talked about how pH would change the state of an R group. So if you had a very low pH, your acidic functional group would no longer have that negative charge. If we go lower pH, all of a sudden that acidic group had that hydrogen attached, so you're going to break salt bridges or whatever it may be. So anyways, just like we talked about how enzymes have evolved to be at a certain temperature and be optimally effective at that temperature, they've actually evolved to be at an optimal pH as well. Physiological pH, we say, is 7. I think more specifically, you could say 7.4 is kind of the magical number. But yeah, around pH 7 is the physiological pH. That is the pH of most of your cells in your body. It actually turns out like a lung cell is slightly more acidic than your blood cells, for example. So the optimal pH of your lung enzymes might be slightly different than elsewhere. But again, roughly speaking, that 7, or more specifically 7.4 number, is what we consider to be a physiological pH. Okay, and just like we saw before, if an enzyme's evolved to operate at this maximally effective optimal pH, anything higher or lower than that, you'll see a decrease in enzyme activity or a decrease in that reaction rate. Okay. Another example of, okay, so here we go. It is optimal pH is about 7.4. Another example, I talked about how your lungs are slightly more acidic. Your stomach is obviously an incredibly acidic environment. Okay. So the optimal pH for enzymes that are active in your stomach is actually more like 1 to 2. So very, very low pH. So you can see that different enzymes, depending on where they were, and I don't want you guys to like have this list memorized or anything like that, but it is just kind of interesting. Different enzymes, depending on where they've evolved to be located in the body, might have a different optimal pH. Your liver, it seems, is a fairly basic environment. Okay, so now effects. So we talked about effects of temperature. Essentially, all enzymes have an optimal pH that's 37 degrees Celsius for humans. Talked about how pH affects the enzyme activity. Every enzyme has an optimal pH. Anything different from that will mess it up. Enzyme concentration, the effect that that has. Increasing the enzyme concentration will always increase reaction rate. Okay, because you've just increased the amount of catalyst you have. So there is a linear correlation between increasing enzyme concentration and increasing enzyme reaction rates. You've just increased the amount of enzyme present that the substrate can bind to. Changing substrate concentration is a slightly more complicated thing. Okay, so if we look at this graph here and we focus just kind of on this front part here, you see at first, increasing substrate concentration increases the rate of a reaction, right? Which is kind of what we would expect. However, then you kind of reach this point, this plateau, where adding more substrate does not increase the rate of a reaction. It plateaus such that if you double the substrate concentration again, you don't actually increase the rate of the reaction. This at this point is what we'd call like the saturation point. Okay, and the whole idea is, remember that you have to form, you go from being an enzyme, the substrate joins, now you're the enzyme substrate complex, then you're the enzyme product complex before the product pops off, and you're back to the enzyme. These two steps right here are the slowest, always. So essentially there's a point where all of your enzyme is in enzyme substrate or enzyme product complex. And so even adding more substrate isn't going to increase the reaction rate. So at this point here, this is the point where all enzyme is ES or EP, right? Everything is stuck in this step right here, which is why adding more substrate doesn't actually increase the rate of the reaction anymore. Okay, so this is the effect on substrate concentration. At first, increasing the concentration of substrate will increase reaction rate, but then you're going to hit a point where it plateaus, and having increasing substrate concentration does not have any effect. Okay, so sucrase has an optimal temperature of 37 degrees Celsius and an optimal pH of 6.2. Determine the effects of the following on the rate of its reaction. Okay, increasing the concentration of sucrase. Increasing the concentration of enzyme will always increase the rate of a reaction. However, what if we change the pH to pH of 4? Anything greater or less than 6.2 is going to decrease the rate of the reaction. Likewise, sucrase has an optimal temperature of 37 degrees Celsius. If we jack that up to 70 degrees Celsius, we would decrease or maybe even outright kill the rate of that reaction at that point, right? So anything other than 37 degrees will decrease the rate of a reaction. Okay.